Today we are going to start with the topic that is the neuron. So first of all, let us know a little basics about the neuron. What exactly neuron is or neural cell. So we all know that the whole human body is made up of different kind of cells and which will aggregate to form the tissues, and then those tissues will aggregate to form the organ. Now different organs, they work cooperatively with each other to form organ system, right? Now whatever the part is there in a human body, each and every part of the human body is being formed by cells. In the same way, there is a system in our body, that is the nervous system, which is going to control other organs and other systems in our body for their proper functioning, right? So all that nervous system is being formed by cells also. So the cells which are forming, which are composing this, the whole nervous system is what we call the neural cell or the neuron. Okay, neuron. Neuron. So neuron, they have got this title that they are the structural and functional unit of the nervous system. They are the structural and functional unit of the nervous system. What that actually means? Structural unit, that means the whole structure of the nervous system is being formed by this. This is the unit. That means the neurons will, a lot of neuron cells, a lot of neurons will aggregate to form the nervous system. Function unit, that means the nervous system, the function of the nervous system. Each and every function of that nervous system is being done by a single neuron. Okay? So today's topic is basically about the structure of the neuron or the neural cell. So let's get started with the structure of neuron. The structure of neuron is similar to that of other cells in our body, right? But there are certain kind of modifications in this cell which is making it a little kind of special. Okay, so what are those modifications? Suppose this is a general cell, okay, with a nucleus. Okay, now in case of neuron, what will happen? This cytoplasm, the cell body of the neuron or the neural cell will have some kinds of extensions. Okay, now let me draw some extensions. Suppose these are the extensions of the cell body or the cytoplasm. Okay, so in the neuron cells, these kinds of extensions will be there, and these extensions are what we call neurites. Neurites. Okay. Now neurites is a term, is a collective term for dendrites and axons. That means it is a collective term for dendrites and axons. Okay. That means dendrites, axon. दोनों को मिला के we call it neurites. Okay. Now what is dendrite and what is axon? There is a difference. That's why they have got different names. So see, out of all these neurites, out of all these extensions, one of the extension will be there which will extend longer than the other extension. Suppose this extension is extending much longer than the other extensions. Okay, so this extension which has extended longer than the others will get a name axon. Axon. Whereas all the other neurites now here will be termed as dendrites. Clear? Now at the end of the dendrite as well as at the end of the axon, that means at their terminal point, there will be the formation of branches. That means they will end in a branching form. These are dendrites, so all dendrites will end with some kind of branching. In the same way, axon is going to be branched again, going to form branches. So how it will form branches? It will form the branches, but at the end of the branches, there will be a formation of a button-like structure. Okay, button-like structure. So I have told you what are axon, what are dendrites. Now we will study or we will learn about the basic things about the whole neural cell, that what is exactly present within the neural cell, about the axon separately, about this terminal part of the axon separately. So as we can see, I have drawn a different structure of neuron, now a proper structure of neuron, which you will get in your books. Okay. Now I have labeled the diagram here. These are the dendrites. This is the axon. They will collectively known as the neurites. Now this part of the neurite or the neural cell, neuron or the neural cell is the cytoplasm as usual, and the body of the neural cell is known as the cell body or the cyton. I have written it here, cyton. Now we will talk about this, okay, the cell body. So similar to that of other cells, the cell body will contain the cytoplasm. Now there will be a lot of different types of organelles which will be present in this cytoplasm. Okay, so each and every organelles which you know will be present here, like the mitochondria, the Golgi apparatus, endoplasmic reticulum, and ribosomes. Everything will be present except, since it's an animal cell, so we know that centriole is an organelle which is present in the animal cell. Okay, neuron is again an animal cell, but in case of neuron, there is a major exception, very important exception. That is the centriole. Centrioles are absent in neurons. They are not present. Okay, so let me write the exception here. Exception. This is very important. Okay, exception is that centrioles are absent. Centrioles are absent in the cytoplasm of neuron cell. Now we know that what is the importance of centriole cell. What is the function of centriole cell? We have studied about the cell division chapter, right? We know that the centriole plays a major role during the cell division of animal cell. So, as the centriole is not present in the neuron or the neural cell, that means this neuron cell will not be able to divide, right? So whenever a question comes that which animal cell is do not have the capability to divide, then your answer should be neuron. Or if they ask the reason, what is the reason behind this? So what will be the answer? What will be the reason? Because there are no centrioles present in the neural cells or the neuron. Okay, so there is a structure in the neuron that is what we call the Neissel's granules. So I have drawn it as granular-like structures in the cytoplasm of the neurons and have labeled it here, the Neissel's granules. So what exactly the Neissel's granules are? Neissel's granules. Okay, Neissel's granules. Hote kya hai Neissel's granules? We know that endoplasmic reticulum is present in the cytoplasm, right? Endoplasmic reticulum is a organelle, right? This endoplasmic reticulum will coil around. Okay, that means it will coil around ribosomes. It will coil around ribosomes to form a different structure. That is what we call the Neissel's granules. Okay. Now, what is the function of the Neissel's granules? As we know, this Neissel's granules, these are the center of protein synthesis. They are the center of protein synthesis in neurons. Clear? So this is what we have talked about, the cytoplasm of the cell body. Now let us talk about the cytoplasm of the dendria. The cytoplasm which is present in the dendria. Similar cytoplasm, right? So everything which is present in the cytoplasm of the cell body is again going to be present in the cytoplasm of the dendria, right? But one more thing I have forgotten to tell you, that is presence of neurofibrils. Neurofibrils are also present. Okay, so these neurofibrils, Neissel's granules, everything, any other organelle which is present here will also be present in the dendrite. Okay, now function of the dendrite. What's the basic function of the dendrite? So dendrite has a major function. What it do? It collects the signal. And it passes it or conducted it in a centripetal form. That means towards the center. Okay, signal को ये लेगा, signal यहाँ पे आएगा. It will gain that signal, it will contain that signal, and then it will conduct that signal towards the center of the cell. That means in the centripetal direction, right? So it's the function of the dendrite. Now let's move to the axon. Okay, starting from the point where the axon is starting. So this part of the cell body from where this axon is getting started. Okay. So this point is what we called axon hillock. Axon hillock. So this axon hillock, that is the starting point of the axon, is the trigger zone. Trigger zone of the neurite cell. Why? Because this is the point where the action potential will be generated, right? Whenever there will be the conduction of the nerve impulse, okay, whenever an impulse comes, whenever the cell will get an impulse, there will be a generation of action potential, which we will study later during that the nerve conduction topic. Within that topic we will study about the whole action potential generation system. So the action potential generates at this point, and that is the reason why this point is known as a trigger zone. Okay, now let's move to the axon part. Now I have told you this part of the neuron is what we call the axon. The point from where it gets started is the axon hillock, or the trigger zone of the neuron. Okay. Now let's talk about the axon particularly. So we know that any cell will have the plasma membrane, right? In case of axon, here the plasma membrane, the membrane of the axon is known as exolemma. Exolemma. Okay. And the cytoplasm of the axon is known as exoplasm. Exoplasm. Okay. Now there is a basic difference between the cytoplasm here and the cytoplasm here. What is the difference? The exoplasm will devoid of two things, that is the Needles granules and the neurofibrils. Neurofibrils. These two are the structures which are not present in the exoplasm, which is not present in the exoplasm. So these two structures are absent here. Okay. Now let's move to the end of the axon. This end of the axon is what we call telodendria. Telodendria. Okay. Whereas these button-like structures, which are the most terminal part of the axon, are known as synaptic lobes. Synaptic lobe. Clear? Now this axon is being covered by a sheet. A sheet which is formed by a layer of phospholipid, like this. Okay. So this structure, this layered structure, is known as myelin sheath. Myelin sheath. Okay. Now what exactly myelin sheath are? Myelin sheath are formed by the squamous cells. Squamous cells. How they are formed with the help of squamous cells? Let me tell you. So suppose this is the squamous cell. Okay. Now this squamous cell will form certain extensions. Extended. Now this extension, it will coil around. It will form turns around the axon to cover it up. Okay. Now let me draw a kind of let me represent it diagrammatically if possible. Suppose this is the squamous cell, it has extended, now it will turn, it will coil around the axon, okay? So starting from here, it is coiling, okay? Much better. This portion. So I have drawn the diagram here. The squamous cell extended, and after extension it got coiled around the axon. So this is the axon, okay, and it has formed a layered structure, right? जैसे ये इसके around घूमने लगा, चक्कर लगाने लगा, it got coiled around it. It formed a layered structure. This layered structure is now known as the myelin sheath. Myelin sheath. Okay, myelin sheath. So we are done with the myelin sheath. Now we can see here if the myelin sheath is covering the neuron. Okay, if it is covering the neuron, then there are certain points where the myelin sheath is absent. Right? Here in this, at this point, these are the regions where the myelin sheath is absent. So these regions where myelin sheaths are absent are known as node of Ranvier. Node of Ranvier, not Ranvier, Ranvier. Okay. So these are the regions where the myelin sheath is not present. Now, what exactly myelin sheath has to do, or what exactly is the function of the myelin sheath? So myelin sheath, wherever this myelin sheath is present, it makes the axon part here non-conducting, or it is performing a function of insulation. Okay, it is acting as an insulator, so that whenever an impulse will come from this point where the myelin sheath is not present, or the node of Ranvier, this impulse will directly conduct it to the another node of Ranvier without passing through this, because it has become insulated now. The impulse cannot be transferred from here, right? So ultimately it helps in reduction of the time for the conduction of the impulse. So faster conduction, it will help in a faster conduction. Okay. Now we are done with the axon part. Now let's move to the telodendria part. I have drawn an enlarged structure of the telodendria. So this part is the axon terminal. This axon terminal is what we call telodendria. And these button-like structures are what we call synaptic node. Synaptic node. Now, what exactly happens in this synaptic node? In these synaptic node, vesicles are present. Vesicles. These vesicles are known as synaptic vesicles, which contain neurotransmitters. So, what are neurotransmitters? These are the synaptic vesicles. In them, neurotransmitters will be present. Okay. So, what exactly are neurotransmitters? They are nothing. They are just a chemical substance which is going to help for the transmission of impulse from one neuron to the other neuron. We know that here this is the axon part. Okay. Axon. To the next, the adjacent neuron, the dendrites of the adjacent neurons will be present here, right? So the impulse which is present in this axon needs to be transmitted from here to this neuron, so that it will conduct that impulse throughout its length, and after that again it will transmit that impulse to the next or the adjacent neurons. And ultimately it will reach the central nervous system. Okay. So for this transmission, this transmission can occur in two ways: electrical and chemical transmission. That we will study later on. So two ways. Electrical impulse will just occur like the impulse will directly transfer from here to here. How? The axon and the dendrite will have a very less space in between them, so it will directly move to here. But in case of chemical synapses or chemical transmission, certain chemicals are there which we have termed neurotransmitters. These neurotransmitters are going to help the neuron to transmit the impulse from here to the next neuron. Okay. Here we are done with the structure of the neuron. Now let's move to the types of neurons.